This is Dan Garcia. Yeah. Okay, so today we're going to have a special guest, Professor Lydia Son. She's a faculty at Mechanical Engineering. And I, and then also Lydia, thought that Lydia presented exactly the same time last year in this class. And she presented a project. She just contacted me because she was looking for a student to work on, to help with their project. And she presented in the class this amazing project called Node Pore Sensing. And since then, we've been actually working together on this because it was so exciting and it was so relevant actually to the class that what we thought of is maybe presenting the same thing again, but then, so that will be about 10 minutes. She's going to tell you about what is this technology and what type of signals are measured. Then we're going to have maybe a 10 to 15 minute discussion of between you is like how we can improve this. And then I'm going to tell you what we've been doing for the last year since then. So it's something different for sure than what we've done so far. But the whole idea is that, can you leverage what you've learned here and how you can take that into a completely different discipline and see what contribution can be made. So, Lydia, thank you.
All right, thank you. Thanks, Mickey. So, as they say, and thus a beautiful friendship was born a year ago. It's been cool, actually. I love it when collaborations come out this way. Is it even recording me right now? Okay. So this technique that, or method that we've developed in the lab, we call it node pore sensing, and it's now become like our bread and butter in the lab for a lot of different projects. And I kind of think of it as a project that keeps on giving, and it really is very interdisciplinary. So let me just sort of give you a background of why we're doing what I will be showing you, what the motivation is. And so I'm in mechanical engineering, never took an ME course my entire life. I actually got my degrees in physics, low temperature physics, and now I'm doing biology. So it's a very strange life I lead. But one thing that I've learned in talking with biologists and doing it myself, doing some biology myself, is there's something called multicolor flow cytometry. So for those of you who haven't had biology or it's been a long time, just know that cells, any kind of cell, will have these proteins that are on the surface. And these proteins pretty much identify, it's like a fingerprint of the cell in many ways. And biologists want to know what those proteins are, and they want to develop, they've tried to, they develop these, what they call fluorophores. They're these dyes that can attach to other proteins that then stick onto the cell. And what they can do is they can sort of incubate, put all these proteins that have these dyes on them into a test tube, put the cells in there, let everything bind to each other, and then they'll run it into this big machine right here. It's called the BD Influx, and it's actually here on campus in Li Ka-shing in one of the core labs. And this is a huge system. It's very complicated. You can see what it has. It has like several different kinds of lasers. They're all shining onto these cells as they flow through. And then there are all these different kinds of detectors to detect the different emissions of the dyes when they're lit up by these lasers. And when you find that you have a hit, you'll find the dye is particular dye is emitting a particular color. Then that cell is the one that we want. It has a particular fingerprint, and you can isolate it. The state-of-the-art system, like the BD Influx, can do 18 colors, which means you can have 18 different kinds of proteins that you can probe. That means you have 18 different colors that you're trying to detect and everything. But it's very complicated because all of these dyes, they're very close to one another in terms of emission spectra. So you can imagine having 18 emission spectra, they're all overlapping with one another. And so there's a very complicated analysis techniques to try to separate these two, all these different emission spectra. There's a loss in sensitivity, and that is you just lose out because you're trying to refine all these spectral emissions. And it just really takes a long time to do your sample prep. So when I say that the scientists will just basically put all these dyes and proteins in a test tube with the cells, It's taken them about almost a year to figure out which dyes to use, what's the right concentration, what are all the controls. And so there's a lot of prep that goes in. And what my lab is trying to do is we want to simplify that. I should mention, like, this BD influx costs a couple $1 million. You need a dedicated technician and various other people to help you out with these systems. But at the same time, they make a really broad impact in medicine and bio. So what my lab has been doing is we're trying to basically do that multicolor flow cytometry on a chip, on a microfluidic chip, and we're trying to do it label-free. That is, we don't want to have any of these proteins that you have to try to figure out how to label the cell and everything. So just to show you sort of the background and sort of how this project has all developed, we used to do something that we call resistive pulse sensing. Okay? And what it consists of is really just a straight microfluidic channel. Okay? And then we have reservoirs, and we have 4 electrodes. And what we're doing is we're doing a 4-terminal measurement to measure the current across the channel. And we also call it POR for historical reasons. But basically, we're doing what they call a resistive pulse measurement, which is you measure the current across the channel in some constant current. And then as the cell passes through, that cell is really an insulator compared to the conducting media that you're flowing through the channel. And so you see a drop-off in current. And then as the cell passes all the way through, then the channel, once again, basically the cell is displacing a certain amount of conducting fluid. And so as you see is that once this cell passes all the way through, this channel once again fills up with fluid, and you see a rise in current. And it turns out that the magnitude of the pulse that you get, how much does the current drop, corresponds to the size of the cell. It's almost a volume-to-volume ratio, cell to channel. And then the pulse width is really what tells us you'll see is it gives you a duration time, how long it takes for the cell to pass all the way through. But in the end, we use that to determine what is that particular sort of, what are all the different proteins that are on the surface of the cell. And the way we do it is that basically we actually do a technique we call functionalization. We actually put proteins down on the surface of the channel, and then as the cell passes through, it can actually interact in a particular way, which means that you would actually have a hit, that the cells want to bind and stick to those proteins. The cell will stay longer in the channel. It won't bind because we're actually flowing at a high enough flow rate, relatively speaking, or because proteins like to just stick to each other in some random fashion, you'll get something that we call non-specific interaction. The cell will take a little bit longer to pass through, but not nearly as much as when it actually makes a perfect hit. So this is the kind of data that we used to get. We used to have 3 different devices, normalized current versus time. And you can see the blank means that there's nothing that we've prepared on the channel. The IgG2A corresponds to, that's our control. We use a particular protein that's almost like the protein that would like to stick to the cell, if that cell has a particular protein, another type of protein, or this is the specific channel. And what we did is we took these murine erythroleukemia cells, they're mice leukemia cells, and we ran them through our device, and you can see sort of all the data. Each one of these pulses corresponds to a cell. They all vary in magnitude because the cells are all vary in size in a general population. And it's actually, even though I say it's a volume-to-volume ratio of what the cell size is to essentially the current, it's a little bit more complicated. You actually have to solve Laplace's equations for all the electric field lines. But in the end, you can determine the cell size distribution. And here's a case where we were looking for what's known as the CD34 protein on the surface of the cell. And you can see here is that when the cells pass through the blank or through the control, they took about 6 milliseconds to pass through this long a channel. But when they passed through the channel that had these particular proteins on the surface, it took much longer to pass through. And that way we know, just by looking at this transit time, or really the time of flight measurement, we know whether or not we have a cell that's actually has a particular protein on the surface. So this was really cool, but then we made this big discovery, which was really neat, in that, in fact, in many ways, these channels are nothing more than like wires. okay, and conducting wires. And what you can do is you can introduce what we call nodes. It's not a great picture here, so let me just sort of switch into this one. You can see these nodes that we've introduced into the channel. And when you do that, you actually, instead of getting just a single pulse as the cell passes through, now the pulse is modulated or divided per the number of segments that you have in between these nodes. So here, these are 2 nodes, and now you can see that the pulse is subdivided. And the reason why this works is because essentially, again, it's like a wire, and the cell is an insulator, and it's passing through. Essentially, it's just the current density is different in the channel versus when it's in the node. So here you can see that the current density is actually greater. It would be greater than if it were in here. Or if you go back to that whole concept of a volume-to-volume ratio, the cell is displacing sort of more per volume of the channel conducting fluid than here. So that's why you see a rise in current whenever the cell passes through. And with this, it's really cool because what it means is that you can basically encode the channel with spatial and temporal information. So here what we did was we put 4 nodes in series, and all we needed to look for were pulses that were subdivided, but with 5 segments, or like these 4 peaks, sub-peaks. And here we're taking a very large channel, and we're measuring everything from 5-micron colloids all the way down to 50-nanometer colloids. Here's just another example where we were actually looking for virus, HIV, in human plasma. And plasma has like all this protein junk in it. And essentially we used that same channel. And you can see the raw data. It looks very noisy. But we know that we have a particle that passed through because it has 4 peaks, 4 sub-peaks in a pulse. And it's something very large. But here, right there, which is in red, it looks like funky noise, but if you just do a low-pass filter, you can extract out those 4 sub-peaks so that you know that it is a particle passing through, and it turns out to be HIV. So what we're doing in terms of just being able to look at all the different types of proteins that are on the surface, we're taking advantage of that by doing something like this. And this is our version of multi-marker screening or multi-flow cytometry without labeling. What we can do is we can print these proteins in between the different nodes into different segments, and then as cells pass through, if they can interact with that particular protein, they'll take longer to pass through than all the other segments in the channel. And so that way, essentially, we can have a very long channel with all these nodes, and we're just sort of looking to see which cell is taking longer to pass through which parts in order to determine whether or not the cell has a particular protein profile. And this is just an example. I'm almost done. But essentially, here's the thing where we've gone up to 4 different proteins that we're looking for. We've printed all the different antibodies or the proteins. We have our control. And this is the type of pulse that we get. You can see it's pretty slow. And Mickey will tell you sort of, or you guys can figure out how we've sped this up. But it becomes a really interesting thing because, you can go as long as you want in many ways in theory, to look at all these different, essentially the fingerprint of the cell. So, like I said, we're playing around right now. We want to sort of expand that, and you can encode spatial and temporal information by playing with the node spacings. And so here's a case where this microfluidic channel has 2 nodes close together, an extra long node, and then 2 nodes far apart. And the pulses can be subdivided now into a high-frequency signal versus a low-frequency signal. So you can play around with FFT and so forth. And this is my last slide, basically just to show you how this whole thing has evolved. We used to have a microfluidic chip with all a bunch of huge preamps, sources, and current voltage sources and everything. We compacted into a small series of PC boards in a box. And now we're working off of a single PC board. So this just competes with that very large system that I showed you at the beginning. So with that, I'll let Mickey.
Can you just mention the application that you do there for actually cell detection and classification?
What the application is for? Oh, okay. So yeah. So when I talked about like the whole idea that you have these cells you want to be able to identify cells with a particular fingerprint of different proteins. What we're doing this for is application-wise for us, it's really to be able to detect different types of cancer cells that can be in your blood or basically in your blood or in your bone marrow. We've been doing some stuff where we actually can take bone marrow samples, and we can actually help people, the doctor diagnose what the particular leukemia type the person has, and so forth. So, okay.
Any questions? Well, first of all.
Yeah.
Any questions?
Yeah. Such a good question. So we can't. We've been working with very diluted samples. But it's always very clear when two cells pass through at the same time, because then, for instance, I'm not wearing the right shirt for this kind of chalkboard. Instead of getting a pulse that should look like this, if two cells pass through, you can get something that looks like this, and so forth. It gets very complicated, and that's one of the vexing things, and one of the things when I approached Mickey was just sort of like, we'd like to be able to handle two cells passing through that, because that obviously will increase our throughput and everything. And so this is where your class gets into play. Great question. It's almost one-to-one. How well do we match with the gold standard flow cytometry or multicolor flow cytometry? And so statistically, we've run some tests, and it's a one-to-one almost. It is, for me, I still think of it as really amazing. We're just doing transit time versus fluorescence detection. Yeah. Oh, how long does it take to do it? So for us, we can just, once we prepare our chip, it does take us 24 hours to prepare the chip and put down the proteins. But then we could just take our cells and just start measuring. If we did, when we compared with flow cytometry, it would take multiple days because we'd have to incubate the cells with the proteins and so forth and do a lot of control testing. So in the end, it's also very fast. Yeah.
How do you actually prepare the chips?
So we do something called soft lithography. So have you taken, was it 140? Wait, this is 140. Which one is this class? 123. Have you done 143? Okay, so that's the microfab course. So we basically have a clean room in the lab, and we actually print chips that have raised structures, and then we pour this polymer on the surface. And it's equivalent to silicone rubber that you get from Home Depot. It's pretty cool stuff. And we basically cure it, and we can cut out molds, whatever we've printed on a silicon wafer, in the form of our microfluidic chip. And then we have already pre-fabricated electrodes on another substrate, which is glass, and we smash the two together. Before then, we actually do a lot of use this thing to just say it's similar to what we call microcontact printing of proteins on the surface. Basically, you just sort of slam down your proteins on this glass surface and hope some stuff sort of sits on there, and then you just align and start measuring. So, yeah.
So does this mean that each chip is a one-time use?
Yeah, it's designed that way. So, like for our experiments, so even if we go up to, I think I did a calculation once, if we go up to like 10 different proteins and we're measuring, so the chip itself costs pennies or less than pennies. It's really the cost of the antibody. So each chip in the end would cost like $1 or $2. Like with the BD influx, if you were to add in all the stuff that you would have to do, it costs thousands of dollars to prepare an experiment. De novo. A great question as well. So if we prep them, because it's proteins, we'll actually put them in the refrigerator until we're ready to start measuring and everything. When we're doing our runs, so we actually, even though it's small number statistics, it's comparable to flow cytometry, where you would need, like we only need about 100 cells to measure before we know an answer statistically. flow cytometry, you would need 15,000, 20,000 cells. We can run our devices up to a couple, like 8 hours or so, but usually it's just a few minutes to do measurements.
Stick, right?
Oh, absolutely. I'm just going to sit in the back.
So Lydia presented this last year, and you guys are taking this class, so you're doing signal processing. And let's say somebody that is they have specialty in their particular area, right, which is biology or biofluidic and so on and so forth. They want to do some signal processing on this, right? They need really to be able to detect, classify cells, right? That's a very specific question they want to answer, right? So they have some ideas of how to do this, and they know how to implement in certain ways. The question is what you could be doing if you want to help, participate, or because here's where I think signal processing makes a big difference. Is that you learn all this theory, you learn a little bit of practice. We teach you communication, we teach you about images, but there's all other disciplines that people have to do process signals, right? And they don't take this class. They don't take 120. They don't take 20. And so the question is where you, as people that took this class, students that took the class, where can you contribute in being able to help this kind of project, right? So the first thing that you should probably be thinking is what is the real problem, right? What are you trying to do? And so I think this has to be it's not just a single-sided thing where somebody presents you with a problem and then you just immediately try to solve given all those details. It's always, you get more benefit if you start querying back. What can you do with your system? What is the limitation? Why are you doing it that way or that way? Can you make it that way, and so on and so forth? There's always needs to be this going back and forth to really understand what is the problem you're trying to solve for. And I assume that, Lydia, the problem is being able to classify these cells and say something about them, right? What their size, whether they're responding to a marker, and so on and so forth. So, given that you presented this kind of work, so how would you actually process this data? What would you actually do? Right?
It looks like there's a lot of piecewise constant parts.
Looks like piecewise constant parts, yeah.
So you could try using a hardware transform.
Oh, okay. So, right, you could use a high-wavelet transform for some reason, but what is the problem you're trying to solve? What are you trying? What are we trying to do here? Yeah.
We're trying to match fingerprints to each type of cell in some kind of cross correlation.
You're getting to realize you're actually getting to the right direction, but what are we trying to measure here? Right? You've got the signal, right? What are we trying to look at? There's two things really that we're trying to look at. Yeah. Right. We want to measure transit times. Right. Transit times. That will give us some information about whether it's interacting with the protein or not. What else? The amplitude, right? And that would indicate the size of the cell, right? So there's two things that we want to measure, right? Two things. And of course, there's lots of issues there, right? There's noise. Who mentioned multiple cells? Yeah, there's multiple cells. There's junk, right, that goes in, right? So, and the question is okay, so how do I measure these things? Right? That's the real question. And then, can you help also, given the fact that you know now what you need to measure? You need to measure the height, and you need to measure the length. Can you now think of what will be the trade-offs in terms of how long the channels should be? Should they be short? Should it be long? Should it be narrow? I guess narrow channel gives you more amplitude, but the problem that limits the size of cells, that can go through. Right? So if your channel is smaller. If you make your channel longer, what could happen? Well, I don't know. That's where you need to talk to the expert in the area and like what happens if you make the channel longer. And in that case, if you make the channel longer, you start losing signal to noise ratio because you just have the amount of resistance you get from the longer line or the amount of water there is within the channel starts dominating over the changes in the resistance that the cell produces. Okay, so there's a trade-off there. Your signal is going to be smaller if the channel is longer. Okay, so let's say you're given just a very simple apparatus where there is just a single channel and the cell goes through. How can you detect it and give those parameters? And let me show you just a few examples just to stimulate things. There you go. So what is a signal and how you detect it, right? So you can think of it as a signal with different standard noise deviation or just smaller signal with the same noise, right? So it would look something like this. So if I gave you the top signal, how would we detect it? The most simple way. Threshold, right? Your threshold, okay? And how do you base your threshold? Actually,
Based on like a ratio to the max.
so it's a little bit more complex. So with that, taking classes on probability and estimation, detection, things like this. 126, 226, and then what's the name of statistical learning theory? 281. Yeah. So there you learn some rules about distributions and how you make choices that would maximize detection, reduce false alarms, miss detection, and so on and so forth. There's criteria of how to pick them. And if you think about this, this is really a distribution, right? There is a chance, a random chance that you'll have a signal that goes above a certain threshold when you have noise. So but eyeballing it, I would say I'll pick some threshold somewhere in the middle, right? But you can actually say something more informative about this. And if you want to learn about that, the probability classes, the random processes, statistical signal processing 225A as well, will teach you a lot about that too. Okay? So how to actually decide what the threshold is, and what can you say about the ability to detect? How many cases you're going to miss, how many cases you're going to detect, how many cases you're going to detect something that is not real, and the opposite. Okay? So, okay, you pick a threshold. So that's a good one. But what do I do here then? In here. The signal is long enough that you could low pass. Okay, so this is our signal is a rect function, right? So rect function it's kind of a low pass, right? So it's not going to be so much affected if we low pass the signal. And you're right, you're absolutely correct. We can low pass the signal by convolving. And what would be the shape of a low pass? What's the shape of a low pass? A sinc function would be an ideal low pass in time. But what would happen if I just took a perfect rect and convolve it with a sinc? I would kind of distort my signal too, right? Now, what would be the best way to detect it? The maximum. The match. Correlate with what? Correlate with the rect. So in fact, in that case, kind of a moving average, right, of length that is the size of the pulse will give you a maximum, right? Will give you a maximum signal if you integrate over it. And this is called match filtering. You remember match filtering? We kind of did that, right? And so in the match filtering scenario, so actually, add another slide leads to this kind of answer. But here's the thing. If I have a signal like this, I can set a threshold, right, and detect it. If I have a signal like this, I can detect threshold. But does it help me with the threshold to have a longer, right? To have a longer channel. It doesn't really help me, right? But obviously, there's more energy of the signal here than there's here. So something I could do. And so you mentioned low-pass filtering, right? You mentioned low-pass filtering. And low-pass filtering, what would happen after low-pass filtering? I would get something that's kind of more round here, right? Because it would. But what would happen if I would do a moving average with the same size? What would be the result? Well, if you do a moving average with the same size, exactly, so a matched filter.
Kind of like a delta.
Delta? Triangle, right? You convolve a rect with a rect, you get a triangle, right? So here's the thing. When you do a moving average, the signal, if you have a constant signal, it would just add, right? It would multiplying and adding, right? So my signal will add linearly. So if I'm looking for a signal of length 100, right, the result would be, if the signal is magnitude 1, what would be the maximum? 100, right? Because it will kind of integrate over it. You multiply ones with ones and integrate over 100 samples, you'll reach the value of 100. Now noise, when you add noise together, the variances of noise of random variables add. But the standard deviation is square root of that. So noise, as you integrate, increases with square root of the amount of integration, while the signal increases linearly. And if you look at a square root function, as opposed to a linear function, linear always wins. And that's why averaging works. Okay? That's why averaging signals work. Because as you average, noise doesn't increase as fast as actually the signal. Okay? So if I integrate, then the standard deviation of noise, I guess, I don't know if it was 100, I guess, average over 200 samples. This is 100, And if you kind of do a moving average, then standard deviation of noise will be 14.14, whereas the peak signal will be 20. If you compute the signal to noise ratio, you're definitely going to be winning there. Okay? So if you do a moving average type, then ignore right now the red one. Okay? So if I do a moving average on this one, this is the signal I'm going to get out, the blue. Right? Suddenly, your signal-to-noise ratio is more than a factor of 10, even though it was less than 1 before. It was almost 1. Right? So that was a good idea. Now, low-pass filtering is kind of a match filter, but it doesn't match that well. Right? If you do actually a rect, in this case, it matches better. Okay? So that's why when Lydia showed the low-pass filtering result, it did improve. So it's kind of like a match filter for each one of those separate nodes. Now, what the problem is this red thing. And that alludes to your question about what if you have more than one node in a channel. Right? If you have more than one node in a channel, then that will be the result, right, of the moving average. It will be a triangle here and a triangle here, and then it will add together, which would look like a big kind of trapezoid with noise. It's kind of hard to say, are these two cells, or is this one slow cell, or one big cell? What are these? And if you remember the sonar lab, right, the sonar lab, that's exactly what the situation, right? We wanted to detect reflections. We sent small pulses. We didn't have a lot of signal coming back, but we had good resolution. We increased, but we lost the resolution, temporal resolution. We gained signal. And then what we did in order to gain both. To gain both temporal resolution and signal. What did we do? The what? Use the chirp. Why? What does a chirp does? Like what was, and how do you detect the reflections? So what? What was the property of the chirp that we liked? It's linear in frequency. Ah, it's nice. It's a really nice property. That was great. Yeah. It's easier to generate for sure. But what did it help with? Narrow autocorrelation. What does this help with? Detection? So what if I had, what's the problem with this, right? This is just a single, you can think of it as kind of like a single frequency pulse, right? Just a DC, right? And then you do a match filter, and then you get a wide match filter, right? So noise moves your ability to detect where it is in time and kind of gives you low resolution in time. Yeah.
Could we reshape the nodes so we get better signals with narrow autocorrelation?
Interesting. How would you reshape them?
You make them like wider or thinner at certain parts, and that would affect how
What?
wide the signal gets, and you can try to find one that gives you the most.
You're going to look for something that gives you the narrowest autocorrelation. Randomly. Random is good. Is random... Okay, so random works amazingly well for n goes to infinity, for large n. So that's great. Random is a good idea, actually, I have to say. It was a really good idea. But now let's say, well, you have a channel that's certain amount of length. Then you talk to the professional and ask, well, how many nodes we can put like in a channel? Can we put like 1,000? How many? 20 maybe, So then, you start this kind of discussion, trying to understand what are the limitations, right? I want to put like infinite, right, number, because that will be useful, right? I can get like the narrowest possible autocorrelation, but that will be too long. Okay, I can do that over a short amount of time, but can you implement that in PDMS, which is the particular plastic? Well, there's limits on the resolution there. So you start talking, right? But you're doing the signal processing, but it's really important for you to understand what are the limits, so you can affect one side of the system, so you can do a better job from the other side. Okay? So that was a great idea. Random, it's a great idea. Narrow autocorrelation, great idea. So then what do you do? Where do you have these narrow autocorrelation pulses? Where will you look for it? Like I don't know anything about, except I know a chirp. That's a good one. But let's say you don't know anything. What do you do? So you kind of use it in the lab, right? What discipline you would go then to look? Optimization because you want to optimize the narrow autocorrelation, right? But there's so what do we implement in the lab? Lab 1? What was it? What is it called? A sonar. Heard about that. When was the first sonar built? Long time ago, right? There's probably a lot of literature about that, right? These guys already thought about a lot of it. So what you do is you open a book. Seriously, you open a book. You open a book about radar. You open a book communication. You open a book about this type of discipline. Maybe they figured out a lot of stuff, and then it's like, oh wow, oh really? They looked at all this stuff a long time ago. This is a completely different application, but yeah, people have thought about it. So research. Okay? Really, it's what research is all about. You research what people have done. And then you figure out what to do with it. That's engineering, but it's also research. So you open a radar book. And actually, if you look, and you search online too, it's like, well, optimal codes with narrow autocorrelation, pulse compression, these kind of words, right? And then the Wikipedia page pops out. There you go. Barker codes. That's one of the first things that you would actually find out. Okay? Barker codes are binary codes. Okay? There are ones and minus ones. And it turns out that people have figured out optimal pulse compression codes, right? It's a big problem, right? In radar, and people that, governments pay a lot of money to make good radar, right? Like the Army would pay, Air Force, right? So they study this problem like hell. Detection, everything, they studied this. This is a slightly different application, so it has this nuances maybe that doesn't necessarily fit. Obviously, maybe you can think about those concepts. So that's kind of the process that you could go through. So Barker code is an optimal code. This is called Barker 7, but there's basically 7 type bits of information that you send. It's binary. Actually, it's phase shift keying if you think about this. Right, minus plus 1. That's it. So you switch between them with bits, bit length. And the way they use it in radar is they send a pulse, and for a particular length, they send the cosine, and then they invert it over a second length, and then positive, and so on and so forth, according to this particular shape. So it has optimal pulse compression for its particular length. So for length 7, if you look at the autocorrelation, it has 7:1 peak to side lobe ratio. Okay, the length of the width of the main lobe is 1 bit long, and the peak to side lobe ratio is maximized to 7:1. Barker 13 is 13:1. That's the best, and that's the only codes that are known to have the perfect peak to side lobe ratio of their size. So if you had like a 31 length, there are no codes that will have 31:1 ratio. Above 13, they couldn't find any. Okay? There's 31:2, but there's no 31:1. Okay? So it turns out the use for detection synchronization in radar, Wi-Fi. When you send packets, they code it with Barker so you can synchronize and figure out where the packet starts. Okay? So good stuff. All right, let's say, so you go, you open the thing, you say, well, that's great. Maybe I can use Barker codes. What's the issue here? How can you implement that in Node 4?
It's not a digital signal, but your nodes and channels they're spatial and aren't digital like that. If the cell passes a boundary on a node, you'll have like a linear. Like get a perfect right.
Yeah, you don't get a perfect right, but can you get like this plus minus signal? You can't have the negative, right? It's only one-sided. So it turns out if you only have one-sided and you do the autocorrelation, it doesn't look as nice anymore. So we need a way kind of to encode these bits, right? I'm sure there's like hundreds of ways. So if we had more time, I would give you ways to think about it. But we kind of thought of one way to try. And it's also based on what you learned in a class. How do you encode a bit? There's a few ways, right? You can encode it in amplitude, phase, right? Or you can encode it, how is AX25 encoded bits? Or ADSB? The radar, ADSB, the airplane thing. Manchester coding by transitions. Right? So maybe if I encode by transition, so every bit I'm going to have, I mean, what also remember in your back of your head, I want to be able to figure out also the velocity, right? How long it is. Manchester coding is kind of self-clocking, right? You can figure out what the changes. There are always changes. So if you do Manchester coding, if you look at the autocorrelation, it kind of looks like this. So Manchester coding, you still have the perfect. Well, it's actually not a perfect 7-to-1. You've got this negative side lobe. That's the cost of the fact that you do Manchester coding. This one, peak to side lobe ratio outside is 7-to-1. But your code is now twice as long because there's zeros and ones, like there's twice. So there's a cost that you pay for Manchester coding. But still, it's a really nice shape, right? Really nice pulse compression. So, and it's self-clocking. So how do you implement it? You create this node port that result in a shape that gives you Manchester coding Barker 7, for example. So cell goes, it's high, goes into low, high, low, high, low, high, high, low, high, low, low, high, high, low. And if you look at actually the bits, then you'll see that this is exactly what. So this would be, let's see, low to high. This is low to high. And then I think I encoded this as. Okay, so this is high and goes to low. So this is high and goes to low. This is bit 1. And this is high to low. So this is high to low. And this is high to low. So again, high to low. Right? And now it's 2 bit length high. Right? This is high. Anyway, something like that. Oh, in this figure we added one extra, yeah. Anyway. But the autocorrelation looks like this. Is beautiful. So how do you detect velocity? Ooh, I like it. Nice. Nice thinking. You can evolve with a filter bank of match filters. Each one of them is a scaled version, right? You have to account, of course, for amplitude and so on and so forth, such that the result will be the same strength, right? So you have to think a little bit about it, but effectively you look at one of them, and then you look at the result. Now, this is really similar to Doppler radar. So when you send pulses that are pulse compressed, radars are interested in knowing the range, but they're also interested in learning the velocity. And so if you send a random sequence, it has a really nice what's called ambiguity function, which lets you distinguish between the velocity and the range of the airplane that's coming. Because if you have a shift in the Doppler, then the correlation will be low. Okay? Chirp pulses have a terrible ambiguity function because any shift in frequency will effectively be translated in a shift in position because a chirp sweeps the frequencies. So that's a terrible ambiguity function. That's why Barker codes actually use not the chirps necessarily. So we can look at ambiguity function here. This is actual cells passing through a channel that's Barker 7 coded. And then you look, this is time, and this is the different filters. And you would display this as an image. These are the different filters with different scales corresponding to different velocities. And you see around the cell, detection, cell detection. And there's what's called an ambiguity function, which is how well we can distinguish between velocity and position. And you'll see that there's kind of side lobes, right? And the side lobes, we might mistake them for being either cells or, we can, there's all sorts of things that can happen because of the side lobes. So you got to watch out. But this is a really nice ambiguity function. It's pretty nice and narrow. So noise will throw you off to the left and right and up and down in terms of ability to estimate that position. Okay? So that's one thing that we've been trying right now, and we implemented this actually very quickly. And we're looking this to measure the velocities as well. Now the thing is, when distinguish between two types of cells coming in, right? That's one way. We went right now for a simpler solution where we code, for example, a Barker 5, and then immediately after, we reverse the Manchester coding. So whatever was low to high, we switch it to high to low. And that will give you a negative correlation. So this part of the channel is encoded with the control. And this part of the other half of the channel is encoded with an antibody. And so when you now do the detection, you'll get always two pulses coming out. And if there's a difference in velocity between them, that means that this probably interacted. That's slower than this one. This is the negative pulse. That's the actual antibody interaction. This is a positive pulse. That's the control. And if they're the same velocities, that means that it's a negative result. It didn't interact. It didn't go slower. So that's one idea, but there's so many ideas that can come up to do this. Any questions? Oh, we're running out of time. All right. Right. This is unpublished. So. But what I wanted to show you really is, these opportunities come all the time. Okay? And it's where the boundaries between discipline I think that's where a lot of the bigger contribution can happen today, especially when we talk about signal processing. There's really kind of when I told you it's a Swiss Army knife. It really is. If you have those skills, you can work in another discipline. Most likely you'll deal with signals. And so you can use these tools and try to probe the systems from one side and to help yourself do the better signal processing. But then think about, how to search for things, research. That's all these opportunities are out there. And I just wanted to give you an example that came out of last year. I was just actually Lydia coming give a lecture in the class. So, but it happens everywhere. And it's really just opening your eyes. For those in particular that want to do PhD in signal processing, I would say look for these type of directions. Where there's areas that don't have that expertise, or maybe you can use your expertise to kind of influence and use things from one discipline to take to another. All right. Friday we have another guest lecture. They're going to talk about computational optics, how to use computation and signal processing to improve optical systems.